children, how are you all? I hope all my dear champions are doing absolutely great. Well, today we are going to start off a new chapter, one that has not been discussed with this batch, and that is molecular basis of inheritance, again carrying 13 marks, and I can guarantee you are getting those 13 out of 13. Why? Because you are a person who is very serious about studies, who are ready to give the 3 hours to me, okay, going to watch this one shot lecture and then solve the questions with me after this session. So are you someone like that? Then let me know, and if at all you like this video, definitely do let me know that, and if at all you have any doubts, similarly put it down in the comment section. I am here to help you out. Listen, I want you to have a pen and your NCERT textbook, nothing else, okay, just your NCERT textbook and a pen. So just go with me, whatever I discuss, just see from where am I discussing all these things, I'll let you know, okay, and with that you will get a clarity that okay, these things are being covered, fine. And if at all you feel something is very important, you can jot it down somewhere, okay. And this session would be fast because obviously the chapter is huge, and this the term for this series is what, basically revision. It's a revision sort of thing, so you should be considering it like that itself, okay. So are we ready? If you are ready, click on the like button and let's go. The first thing, my dear children, when you talk about the DNA, what is the DNA made up of? It is made up of something that is known as nucleotide. Now what is the nucleotide made up of? Bacha, there are 3 components out there, you have to remember it. One is the nitrogenous base, second is the pentose sugar, and third one is the phosphate group. The most important one is the pentose sugar. The pentose sugar means there are 5 carbons in the sugar. Where are the 5 carbons, ma'am? First carbon, second carbon, third carbon, fourth carbon, and the 5th carbon is outside. So this is the pentose sugar. Okay, and over here you see on second carbon there is no OH group, there is only hydrogen group out there. So this is known as deoxyribose sugar. Usually you have an oxygen over here, but that oxygen is removed, that is why it is known as deoxyribose. So the pentose sugar's name is what? Deoxyribose sugar. Now if at all it is RNA, if it is RNA, they have O over here, which is why it becomes just ribose. It becomes ribose sugar. So as of now it is DNA, so it is deoxyribose. And now my dear children, on the first carbon, the nitrogenous base is being attached. This is a nitrogen base which is being attached, and the the bond which is there, what do you call this bond as? This bond is known as N-glycosidic bond. We'll come to that one by one. So as of now, pentose sugar you studied. What is the name of the pentose sugar? In the case of DNA, it is deoxyribose. In the case of RNA, it is just ribose, nothing else. Okay. Now the nitrogenous base are of 2 types. One is purines and other one is pyrimidines. Purines and pyrimidines, okay. The purines are adenine as well as guanine. What is it? Adenine and guanine. And the pyrimidines are thymine as well as cytosine, TC. Take care, okay. TC is what? The pyrimidines out there, fine. These 2 type of things are there. Now going ahead, on the 5th carbon, what is on the first carbon? This is being attached. On the 5th carbon you have a phosphate group being attached, my dear children. So this is the 3rd component. Pentose sugar, nitrogenous base, and the phosphate group also should be there. Then only you can make a nucleotide. And always remember the bond which is over here. So you see a carbon is bonded to what? A phosphate group, but with the help of an oxygen in between. So what do you call this bond as? You call this bond as phosphoester bond. Okay, what do you call this bond? Phosphoester bond. We'll come to all of it slowly. I'm just giving you a hint so that you don't feel like, oh my god, what all I am studying. So 3 components are there. What all are there? Can you put it down in the comment section? Or can you write it somewhere? 3 components: pentose sugar, nitrogenous base, and phosphate group. Okay, agreed? Done? Now going ahead with the second thing, formation of the DNA. How does the formation of DNA happen? We studied the component. The monomer of the DNA is what? Nucleotide. Now nucleotide as such do not form. You have to form a nucleoside first. Tide comes later, side comes first. So nucleoside is nothing but the nitrogenous base plus the sugar. That's all. Phosphate group is not there over here. So this thing without the phosphate group is what? A nucleoside. So what do you find over here? You find the first carbon of the sugar is in contact with the nitrogenous base with a bond which is known as N-glycosidic bond. That is what I was talking about. This bond which is there between this and this, my dear children, this is N-glycosidic bond. Now whenever your nucleoside is being formed, my dear children, initially you used to call this as adenine. But now since this pentose sugar is also there, you will call it deoxyadenosine. Okay, now suppose here it is guanine, but when it combines with pentose sugar, you will call it deoxyguanosine. Okay, that is how you call it. So deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. That is for adenine, guanine and cytosine and also thymine. Now, nucleotide formation, how does that happen? Okay, nucleoside is being formed, but how does nucleotide formation happens? Basically, a phosphate group comes and attaches to the 5th carbon of the nucleoside. Via which bond? I told you the bond which is known as phosphoester bond. And when a phosphoester bond is formed, my dear children, H2O is being released. So here also H2O is being released. You don't have to go in that depth. But anyway, this is known as phosphoester bond. Now, so this one nucleotide is formed. Now we want to form multiple nucleotides to come together to form the DNA. How can that happen? So look over here carefully. 5th carbon, phosphate group is attached. Now this phosphate group is attached to the 3rd carbon also of another nucleotide. And here also same thing is happening. Here also there is carbon and oxygen and then phosphate. So can I call this bond again as phosphoester bond? So here also phosphoester bond, here also phosphoester bond, right? Both the places. So can I call it now phosphodiester bond, because 2 places it is happening? I can call this. So if at all you are asked this question that 3rd carbon of one nucleotide or one pentose sugar is attached to the 5th carbon of another nucleotide through a phosphate group, what do you call this bond as? Phosphodiester bond is what you call this bond. Now over here that is being done. Similarly, when you go up, suppose this particular phosphate has no one to join. There is no nucleotide at the above. Then the 5' end will remain like this, will remain the same. So this is known as the 5' end. Similarly, over here the 3rd carbon which is there, it is having OH. It is not attaching to any phosphate group. It is just having OH. It is not getting attached to any phosphate group out there. Then what do you call this end as? This is known as 3' end. Okay, this will be known as 3' end. So whatever, when we study about the DNA, we have seen that there is a 5' end and there is a 3' end. What do you mean by that? You mean this only, that there is no other nucleotide to attach over here. Similarly, there is no other nucleotide to attach over here. Now, what about the another strand which is running parallelly? It does not run direct parallel, it runs anti-parallel, my dear children. That means, if over here it is 5' and here it is 3', then the ulta would be happening over here. Here, opposite side, it will be 5' and here it will be 3'. Why, ma'am? Because, my dear children, if you keep it the same thing, then for example, if you see, this is 5' to 3', right? And over here you have the nitrogenous bases on this side. Now again, if you keep it like this, 5' to 3', the nitrogenous bases would be like this. I don't want it to go like that side. I want it to be inside. I want the nitrogenous base to be inside. So for that, what will I do? I will just make it run anti-parallelly. So it is in 3' to 5' direction and the nitrogenous base are inside. Why do I need to do that? So that these 2 strands remain together. The nitrogenous bases on both these strands, my dear children, are bonded to each other. And I am going to talk about that right now. So over here, the bonds which are there, I told you purines, there are 2 of them, that is A and G, AG, OG, right? And in pyrimidines, there are 2 of them, thymine and cytosine, right? Adenine always binds with thymine. That means, if at all this side it is adenine, on the other side it will be what? Thymine. If this side there is guanosine, on the other side it will be always cytosine. So adenine and thymine has 2 bonds between them. This is what? Hydrogen bond. Guanine and cytosine has 3 bonds between them. This is also hydrogen bonds. Guanine and cytosine has 3 bonds. Maybe because they look so similar, they are very close to each other. G and C look so similar, right? So over here, T are not that similar, which is why they just have 2 bonds out there. Can you remember? So as of now, we have studied about 3 bonds. N-glycosidic bond between pentose sugar and nitrogenous base. Phosphodiester bond between 2 nucleotides, phosphate group and things like that. Okay. And then hydrogen bonds as well. Clear? Crystal clear? Done? Dusted? Okay. Now, I'll go ahead. Polynucleotide chain formation. Nucleotides link via 3,5 phosphodiester bonds. So how does nucleotide link to each other? Via 3 and 5 phosphodiester bond. Done. Backbone. What is a backbone of DNA if you are being asked? It is formed by sugar phosphate units with nitrogenous bases projecting inwards. So what is a backbone if you are asked is what? The pentose sugar as well as the phosphate group. The pentose sugar as well as the phosphate group, that is something that forms the backbone, as well as the nitrogen. If you use the nitrogenous bases which you see, they are projecting inwards. Now if you compare some other thing, that is RNA versus DNA, what will happen? What is different in the case of RNA? First of all, I told you, in the case of DNA, if at all you are looking at it carefully, here the second carbon which is there, it does not have an OH group, it just have an H group, which is why it is not being mentioned over here. But in the case of RNA, it has an OH group over here. And not just that, my dear children, if at all you are asking about the nitrogenous basis, RNA which is there, it does not have thymine. It does not have thymine. Then ma'am, what does it have? It has uracil. It has uracil on the place of thymine. So adenine binds with uracil instead of thymine. So how many pyrimidines now do you have in total? T, C and U. In the case of RNA, you have U instead of T. In the case of DNA, you have T instead of U. Got it? So I hope this is clear, crystal clear, done, dusted. Let's go ahead. Double helix model. Now what is double helix model? If at all you are asked, tell about the double helix model, and this is a 5-marker question as well. So two antiparallel polynucleotide. Polynucleotide means one nucleotide, double nucleotide, triple nucleotide, so many nucleotides, polynucleotide chains come together from 5' to 3' polarity and 3' to 5' polarity with a sugar phosphate backbone, and bases projecting what? Inwards. Base pairing occurs. Base pairing means all the nitrogen bases, they are pairing with each other, that is known as base pair. Base pairing occurs via hydrogen bonds, A with T, 2 bonds, and G with C, 3 bonds, ensuring uniform strand distance out there. Okay, now right-handed helix is there, that means they are circling in a right-handed fashion. So right-handed helix is there with a pitch of 3.4 nanometer. Ma'am, pitch, what are you talking about? So basically when you talk about the DNA, this whole thing from here to here is considered as one helix. Now my dear children, over here, what is the distance? It is 3.4 nanometer, and if you are writing in Armstrong, because that is also being asked, it is known as 34 Armstrong. Either way. Now I am telling you, in this one helix there are 10 base pairs. How many base pairs? 10 base pairs. So in between the 10 base pairs, what is the distance? What is the distance over here? What is the distance over here? What is the distance over here? The total distance is 3.4 nanometer, and there are 10 base pairs. Then what is the distance between each of them? It will be 0.34 nanometer. Or if at all you are talking about Armstrong, it would be what? 3.4 Armstrong. Always remember, Armstrong and nanometer is totally different. Okay. So in your textbook, if it is nanometer, then please study that. 3.4 nanometer is over here, and 0.34 nanometer is the distance between each of the bases. Now base pair stacking and H bonds provide structural stability. Base pair stacking one over the other, and hydrogen bonds provide what? Structural stability to it as well. Done. This is your double helix model, my dear children. Will you be able to remember it? I hope you do. Now going ahead with the next thing. So have they spoke? Okay. Anyways. Now discoveries. So we studied the structure, basic structure, and before I go ahead, Okay, wait. There was one point, I think we are missing that one point. Yeah, also thymine which is there. Thymine in the case of DNA, you call this also as 5-methyl uracil. What do you call this as? 5-methyl uracil. Because initially, my dear children, you just had RNA. And RNA had uracil. On this uracil's 5th carbon position, my dear children, you added one methyl group, and that's how it transformed into what? It transformed into something that is known as thymine. Okay, it transformed into thymine. So that is how thymine is being formed in the case of DNA. So 5-methyl uracil is nothing but thymine itself. Please do not forget it. That's all. So we studied the structure, whole thing is clear. And now in your book, there's a paragraph where it is speaking about what you call the discoveries. This is given just before the explanation of the double helix model. So discoveries, the first discovery is in the year 1869. Frederick Mischer, okay, he was a Swiss physician. He identified there is some acidic substance in the cell's nuclei, which he called as nuclein. Can you guess what is this acidic substance? It is none other than your DNA, my dear children. Why do you consider DNA to be acidic? Because I told you it has a phosphate group, and you know this phosphate group is attached with an oxygen, and this oxygen, my dear children, has the negative charge. So if at all you do not want to go in depth, just understand the phosphate group is the one that in some ways is giving what? The negative nature or the acidic nature to the DNA molecule. So Frederick Mischer, while he was eating the mixture, found out that there is some acidic substance in the cell's nuclei which he called as nuclein in the year 1869. Now going ahead, 1953, after so many years, my dear children, two people came here, James Watson and Francis Crick. They said that there is DNA which is having double helix model. And how did they see that? They saw Maurice Wilkins and Rosalind Franklin's X-ray diffraction data. So X-ray diffraction data was being studied, and then later on said, oh my God, I think the DNA has a double helical structure. Who said that? James Watson and Francis Crick. Watson and Crick said that DNA has a double helix model, with whose data? With Maurice Wilkins and Rosalind Franklin's data, X-ray diffraction data in the year 1953. Now, if you go ahead, one more thing is being mentioned about Chargaff. Chargaff, my dear children. So Chargaff said that their model incorporated Erwin Chargaff's finding that in double-stranded DNA, the ratios of adenine to thymine and guanine to cytosine are constant and equal to, and ma'am, I didn't understand anything. Listen, I told you adenine is going to base pair with thymine, guanine is going to base pair with cytosine. Now you tell me, if 50 adenine is there, obviously 50 thymine will also be there. Agreed? If 50 guanine is there, 50 cytosine also will be there. Agreed? So here it is said, again Erwin Chargaff said that the ratio of adenine to thymine means 50 by 50, adenine to thymine, ratio of adenine to thymine and guanine to cytosine. So guanine is what? Again 50, cytosine is also 50, okay, will be equal to what? 1. Isn't it equal to 1? They would be equal to 1, right, my dear children, because both of them are going to be same, okay. Or you can just modify this term and say one more very important thing, which is that purines is equals to pyrimidines. Purines is equals to pyrimidines. That means A and G is equals to T and C. Why? Whatever number of A is there, that means number of T will be there. Whatever number of G is there, that means number of C will be there anyways. So purines is equals to pyrimidines. That can also be said. So Erwin Chargaff was the person who said this, and with the help of that also, what James Watson and Francis Crick were able to deduce what? The double helical structure, that probably this is how things are working out there. So 1869 them, okay, Mischer, he's a cis physician, and then 1953 James Watson and Francis Crick, and then Erwin Chargaff. Okay, now what is the dimension of the nucleus? Now we are going ahead. I know the central dogma is missed over here. I'll tell you later on whenever it makes sense. Okay, so just wait for some time. Now, my dear children, over here, what is the dimension of the nucleus? It is 10 to the power minus 6 meters. 10 to the power minus 6 meters means it's very small. And now, what is the length of our DNA, my dear children? It is 6.6 into 10 to the power 9 base pairs. Ma'am, I didn't understand. In meters, can you tell us? So, in meters, if you do, if 6.6 into 10 to the power 9 base pairs are there, and in between each of them 0.34 nanometer is the distance. Okay, 0.34 nanometer is the distance between each of them. And if I want to convert into meters, my dear children, I will do 10 raised to the power minus 9. Okay, then the thing I get is 2.2 meters. What will I get? 2.2 meters. So what is the length of our DNA? It is 2.2 meters. And what is the dimension of our nucleus? Just 10 raised to the power minus 6 meters. Do you think you can fit this big thing over here? Well, I don't think, but we do. How? We make it compact. We make the DNA really, really compact. How? With the help of what? With the help of something that are known as histone proteins. And now this is also one very important question. The diploid content of base pairs in our body is what? 6.6 into 10 to the power 9 base pairs. What is the haploid content? For example, if we are talking about sperm, if we are talking about egg, what will be the content of base pairs out there? It will be half of it, which is 3.3 into 10 to the power 9 base pairs. 3.3 into 10 to the power 9 base pairs. Always remember it. So if at all you are asked secondary oocyte, what is the number of base pairs out there? It is 3.3 into 10 to the power 9 base pairs. Primary oocyte has not completed meiotic division, so it is just diploid, so it will be 6.6 into 10 to the power 9 base pair. Always remember, these type of questions just definitely comes in. Okay, even the Chargaff's rule, that is also very important. But anyways, okay, so how do we do it? By compacting it with the help of histone proteins. Now what is a histone protein? So basically, 8 histone proteins come together to form a globe-like structure. Okay, 8 histone proteins come together. So this is known as octamer of histone, since 8 histone proteins are coming together. And this is highly rich in positively charged amino acid. Now, 2 positively charged amino acids. My dear children, please understand, why do you need positively charged amino acids over here? DNA is what? Negatively charged. If you give negatively charged amino acid, negative comes, it will repel. So you don't want that, which is why you need positively charged amino acids, and the names are also mentioned, arginine and lysine, AL or La. AL or La, either way you can remember it. Arginine and lysine are the 2 positively charged amino acids which are found richly in the histone proteins. Now 8 of them came together to form a globe-like structure like this, the green one that you see. And around it, what happened? The DNA which is there, it is going around twice. It is moving around twice. It moves around twice. So roughly how many base pairs of DNA are being moving around? It's almost 200 base pairs of DNA which are moving around. Always remember, it moves around twice and 200. Easy, no? Okay. Now, always remember, okay, so this is one histone protein plus the DNA. What do you call this whole structure now? You call this whole structure now as nucleosome. Nucleosome. This is nucleosome. Now, over here, once the DNA comes out like this, there is one other histone protein as well, which is known as H1 protein. Okay, H1 histone protein. Now, there will be another nucleosome as well. Okay, where again it will move twice, and the same thing will go on. And when it go on like this, my dear children, what will happen? What is going to be formed? It forms something like this. So there is a bead. So same thing goes on. So it looks like a string on which there are so many beads. So this is known as bead on a string structure. And what do you call the whole thing when it becomes compact? You call this as chromatin, my dear children. This is known as chromatin. Chromatin later condenses to form what? Chromosome. So initially the DNA is there. Now histone proteins are there. Now they are compact. This is known as chromatin. Chromatin later condenses more to form what? The chromosomes. And over here between 2 nucleosomes, what do you have? One H1 histone protein as well. Please remember it. If at all you are said that the H1 histone protein is very near to both the nucleosomes, that means what? This particular chromatin is very much close to each other. The chromatin is very much condensed. Okay, if at all it is said it is far away from each other, 2 nucleosomes, then that means it is loosely packed. Now, coming on to that particular statement, we'll go ahead and see this. So this thing that you see over here, it looks like a bead, right? On a string. Now, as I told you, nucleosome unit comes together to give a bead on a string structure which is known as what? Chromatin. If at all you are not able to see it, I'll tell you, it is the same thing, bead on a string structure which is known as chromatin. Now, one very important thing, I told about histone proteins, 8 histones are there. Apart from that, H1 histone is also there, a lot of things like that. Now, there are some non-histone chromosomal proteins as well. Please remember, there are some non-histone chromosomal proteins which are also required for formation of chromatin. Now there are 2 types of chromatin. One is euchromatin and the other one is heterochromatin. What do you mean by euchromatin? Over here, my dear children, the DNA which is moving twice, here also moving twice, here also moving twice. You see the distance? It's quite far away from each other, right? They are quite far away from each other, so there is enough space out here. So that means they will take up stain, but it will stain very, very light. Okay, first of all, the first feature that you got to know is what? It is loosely packed and that it stains light. And what do you call this as? This is known as euchromatin. Okay, we'll come to this. Over here, they are very close to each other, which is why there is less space, or can I say it is densely packed? Yes. And when it is densely packed, it will take up more stain. It will be dark in color. The stain would be dark in color. Now, if we talk about a process of transcription, what happens over here? What is transcription? First of all, let me tell you that. So, in our body, we know DNA is there. We have studied in principles of inheritance that DNA has genes, segments that codes for protein, right? So, how does it code for protein? The DNA which is there, it is going to give us information or passes information into something that is known as RNA. And this process is known as transcription. Later, this RNA's message, whatever message is there in this RNA, it is translated to form a protein. So, the next process that happens is what? Translation. So, first transcription to form an RNA, and then RNA into protein is known as translation. Now, for transcription to happen, to make it an RNA, you need certain enzymes, and those enzymes need certain space to come and bind over here and form the RNA. So over here it is easily possible. Transcriptionally they are very active, transcription can take place. But over here, do you see any spaces? So do you think transcription can happen? No. So it is transcriptionally inactive. Heterochromatin is transcriptionally inactive. Euchromatin is transcriptionally active. You can remember like that. I know that's too much of drama, but that's the way you remember things. I know that as well. So now even 17 degrees Celsius is making me feel very, very hot. So what do you think? What can be done? Nothing. So that's super energetic I am as of now while explaining this. My dear children, this chapter is super, super important. So please pay attention to me. If you lose the concentration, you will lose everything. So this is euchromatin, this is heterochromatin, this is transcriptionally inactive. So nucleosome is also done. Our DNA structure is done. The the dimension of the nucleus is what? 10 to the power minus 6 meters. 10 to the power minus 6 meters. What is our base pair? 6.6 into 10 to the power 9 base pairs. 6.6 into 10 to the power 9 base pairs. Haploid is 3.3 into 10 to the power 9 base pairs. If you want to convert into meters, then 2.2 meters the long DNA is there, which is why we need to arrange them in a compact way. Now my dear children, next thing comes up, the search for genetic material. Okay, DNA and everything has been taught and you got it already. Now people were behind, what is a genetic material? Is it DNA? Is it actually the DNA that everyone is talking about? Or is it something else? Is it some protein that is genetic material? What is something that defines each and every part of a body? What is something that is so very important in the body? So for that, experiments were started off. And one such experiment was done by the famous, very famous Frederick Griffith. And this was done in the year 1928. Remember, in 1953, James Watson and Francis Crick came up with the structure of DNA. Before that, okay, way before that, some people were in a quest to find what is the genetic material out there. And he did an experiment using a bacteria that is Streptococcus pneumoniae, which causes pneumonia in organism. It's a bacteria Streptococcus pneumoniae. Okay, now there were two strains of Streptococcus pneumoniae. Strains are in varieties. One was S strain, smooth strain, and one was R strain, that is known as rough strain. So whichever thing looks good might not be actually good. And this is the best example of it. The smooth strain, why is it smooth? Because it has an extra mucus coating. Okay, it has an extra mucus coating. This mucus is made up of polysaccharide. That means it is made up of sugar. Okay, it is made up of sugar. So the smooth strain has a mucus coating which is made up of a carbohydrate. Okay, or a sugar as in not tasty sugar as you feel so. It's just carbohydrate. You also call this as polysaccharide. But the problem with this coating is that it makes the strain virulent. Virulent means it is toxic. If at all some organism is affected with this strain, they will definitely die. It is virulent in nature. Whereas the R strain, which is rough, does not have this thing, and which is why it was what? Non-virulent. It was not causing any trouble out there. Now it's experiment time. So listen, Lily is our mouse. So in Lily, we put the live R strain, the living R strain. When we put it, Lily was totally fine. The mouse lived. Then we put live S strain. When we put it, mouse died. Okay, mouse died. Now we take Silly. Silly is the next mouse which is there. Now on Silly's body, we are going to put heat-killed S strain. We got to know one thing, that live S strain definitely kills the mice. So we treated the S strain with heat. Okay, such that it damages some part of it, and then my dear children, then we treated it. And then we got to know that, okay, Silly is totally fine, it's totally good. The heat-killed S strain does not work. It is not virulent form. Now, Griffith was like, no, I want to do one more experiment. He took the heat-killed S strain, same like he took here, and he took a live R strain which was not causing any trouble. So both of them individually, they were not causing any trouble. But when they were mixed, the Silly mouse also died. Silly mouse died, Lily mouse died. Now it was a problem. He was not understanding anything. So it's like something is there. Okay, some problem is there. And then he got to see that there is live S strain in the group 4. In the group 4 experiment on silly, in silly mice he found that there was live S strain, but he didn't put live S strain anywhere. He put heat killed S strain and he put live R strain, which was not causing any trouble. Then from where is the live S strain being formed? That was the huge problem out there. So he got to understand that some transforming principle, okay, something was getting transferred from the heat killed S strain to the live R strain, making it transform into what? Live S strain. So that has enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. So there was something getting transferred over here, which is why the live R strain was able to make that mucus coating by itself and transform it itself into what? The live S strain. So he didn't understand what was that thing. He called this as transforming principle. When did this happen? In the year 1928. Griffith did it. Frederick Griffith did it. And this was the first experiment for the search of genetic material. And then came other guys, Oswald Avery, Colin MacLeod and Maclean McCarthy. These three guys, okay, in the year 1933 to 44, almost for 10 years they did the experiment. 11 years, okay. They did the experiment as in to see what was the thing that was transforming them. So in their experiment, they used three enzymes. One is protease that basically breaks down proteins. One is RNase that breaks down RNA. And one is DNase that breaks down DNA. And then his experiment, they did this particular thing. That is, the heat-killed S cells were taken, the ones that does not cause any trouble, right? And then they were treated with proteinase actually. So when it is treated with proteinase, the proteins are going to be killed in the heat-killed S strain. Now the heat-killed S strain is mixed with R cells. Okay, it is mixed with R cells and still they were able to see that S cells were appearing. That means transformation was still happening. Even though I damaged all the proteins, transformation was somehow happening. Same thing, he took the S strain, heat killed S strain, treated with RNase, and my dear children over here, S cells were still appearing out there. Again a problem. Then my dear children, again what was being done over here? So the heat killed S cells were taken and now it was treated with DNase. Okay, DNase. Now the DNA was getting damaged and after that when he mixed it, there was no S cells that were appearing. That means probably it is the DNA that is getting transferred from the heat killed S strain to the R strain, which makes it virulent, right? That is what we conclude. They did the experiment. They were actually very brainy people and others should have believed them. But the problem is that no one believed them. So many people believed them, but not so many. There were so many people yet to be believing them. That is why we needed one more experiment to counter-proof it. So isolated components, what they did? They isolated components, proteins, DNA, RNA from heat killed S strain, introduced each component to, okay, I explained all of this thing. Anyways, so DNase treatment inhibited their transformation. So you can be asked, okay, if I used proteinase, will you see any change? So you should know, no change. RNase, no change. DNase, yes, change will be there. Okay, so this is what, this is the type of questions that you can get. Now the last experiment to prove this was done by Alfred Hershey and Martha Chase. Hershey and Chase experiment in the year 1952. This finally proved that DNA is the genetic material. And what did they do to prove it? So over here, they took two bacteriophage. Bacteriophage are viruses, they are not bacteria. Please remember, they are not bacteria, they are the ones that infects the bacteria. So here is the bacteria which is known as E. coli. Over here also you have E. coli. Now he used two radioactive molecules. One is radioactive P32 atom and the other one is radioactive S35 atom. Now sulfur is a component of protein, which is why wherever the protein was there the radioactivity will be there. So the protein is basically over here in the bacteriophage capsule, in the outer part of the bacteriophage. Whereas when you talk about phosphorus is a part of DNA. Phosphate group, it's a is a part of what? DNA. So it was directly going and radioactive labeling what? The DNA out there. Now he did the experiment, he made it go infect this, he made it go infect this. So the usual trend that happens is that, my dear children, it would definitely pass the genetic material. Okay, we as of now do not know what's the genetic material, but anyway it will pass the genetic material. So either protein or the DNA or anything else, right? But here over there, it was passing something and leaving the other materials out there. The capsule out there was still there. Okay, so infection was done After that, blending was done to remove anything that was attached to it. So blending was also done. After that, centrifugation was done, where it is spinned at high level, my dear, high speed. When centrifugation was done, it was found that always there is one theory, that is the pellet will be coming down in the tube, okay, the one that has high density will be coming down, and whatever rest over things are there in the liquid form is known as supernatant, that floats around. So over here, when the supernatant was seen, it was seen that it was radioactive, okay, and always remember, supernatant always contains the proteins, and the pellet will contain what? The genetic material, because pellet has the cell, and the cell will be having the genetic material, my dear children, right? The cell will be having the genetic material. So pellet which was there over here, it didn't show any radioactivity. That means if there is no radioactivity in the pellet, that means the S35 which was used, my dear children, it is going into the proteins, and the proteins were definitely not being transferred to the cells. Proteins were definitely not being transferred to the cells, otherwise it would have been radioactive over here. But on the other hand, in this case, radioactivity was found in the cells. And here, what was radioactive? What was made to be radioactively labeled, my dear children? It was the DNA which was made to be radioactively labeled. So with that, they got into conclusion that, okay, so DNA is something that is getting transferred, which is why I am able to see it over here. You get it? So this was the experiment of Alfred Hershey and Martha Chase, and with that they finally proved that DNA is what? The genetic material in the year 1952. Okay, always remember S35. So S and 35, 5 looks like S, right? So you can remember like that, and P32. Okay, going ahead. What is the first genetic material? So we studied about, okay, the discovery of the genetic material was being done, that is DNA. But ma'am, what was actually the first genetic material? Was it actually the DNA? No, the answer is no, it was not DNA, it was what? RNA. RNA was the first genetic material. Was it only acting as a genetic material or anything else as well? The answer to this question is, it was also acting like a catalyst. What do you mean by catalyst? Something that can speed up a process. That means it can act like an enzyme, a sort of an enzyme. It is able to speed up a process. For example, I want to convert A to B, I will use a catalyst for helping me in that. So RNA was just not just a genetic material but also a catalyst. Is that a problem? The answer is yes. RNA being a catalyst, that means it is reactive and hence very unstable. Something comes up the next day in my body, my dear children, if RNA is a genetic material, if something reactive comes in, obviously I will also react. I have the RNA, not me. The RNA within me will also react to it and will change. And if it changes, my dear children, the proteins that it will make will also change, and that is a huge problem. It cannot alter around like that. I need something stable. So initially, yes, RNA was the first genetic material, agreed to that point, but please understand that DNA is better, and I am going to prove you that here. So this is a question that usually comes, what is the criteria for being a genetic material? First one, it should be something that should replicate, it should be able to replicate. So you have the DNA and you have the RNA. Okay, you have the DNA and you have the RNA, both of them, they can replicate, but DNA does it in a better way. Okay, now capable of self-duplication to ensure what? Genetic continuity, that is why you need it. Stability, chemically and structurally stable to maintain integrity across an organism's life cycle. So this is also a criteria. It should be stable. If it is not stable, it will go on reacting and then things would be problematic. So DNA, if you see, it is more stable due to what? Complementary strands rejoining after heat. The complementary strands, they rejoin fast. Okay, it is not single-stranded mostly. Lack of reactive 2-OH group. I told you the second carbon, they do not have the OH group, and this OH group is very reactive. So RNA has it. That means it is not stable, because it is very reactive. Presence of thymine instead of uracil. Again, thymine is there, which is more stable. So 3 reasons. Complementary base pairing after heating. Second thing, lack of reactive 2-OH. And the presence of thymine in the place of uracil, which makes our DNA more stable. Now, if we talk about mutation potential, for first thing is replication, second thing is what? Stability, third thing is that mutation potential. What do you mean by that? Changes should happen in the DNA. Definitely. Changes should happen in the genetic material. Why? Because if change is not happening, evolution will not happen. So changes should be there, but within a limit. So that is also a criteria. Allows for gradual changes to facilitate evolution. Over here, DNA mutates faster due to what? Instability. RNA mutates faster due to what? Instability. RNA-based virus evolve what? Rapidly. And that is a problem. You do not want rapid evolution to happen. You do not want to mutate faster. And which is why again what? DNA would be better. Now the fourth thing is expression. Whatever code is there, whatever things are there, it should be expressed in the form of Mendelian inheritance. Whatever Mendel said that this trait, this gene is there for this particular trait, it should be showing up like that. Okay, so RNA which is there, it directly codes for protein, but DNA relies on RNA for protein synthesis, I told you. So that is just the drawback which is there, but still, isn't the DNA giving rise RNA? And then, I mean not giving rise, isn't the DNA helping in the formation of RNA, and this RNA is ultimately helping in the formation of protein synthesis as well. So if at all you see, if you compare RNA and DNA, it will be DNA that is best to be considered as what? The genetic material. DNA is preferred as genetic material information storage due to stability, whereas RNA is preferred for transmission. If at all, if you want to transform something, if there is something like that happening out there, you will prefer an RNA if you want faster evolution and things like that. But in all the other cases, as a genetic material, you will use this because the four conditions are matched up over here: replication, stability, mutation potential, and also expression. Please remember, this is a very important question. Now we will have a one-on-one comparison. The sugar, the pentose sugar which is there over here is deoxyribose. It lacks one oxygen at the second carbon position. Ribose, here it has an extra hydroxyl group. Hydroxyl means OH group. Here it is just H, here it is OH. Now it is double stranded mostly. Here it is single stranded. Now nitrogenous bases, thymine is present. Here uracil replaces thymine. Function mainly acts as genetic material. It mainly involves in protein synthesis and what? Transmission. Only one main type of DNA is there, okay, but over here multiple types of RNA is there: mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. We will study about that. Location, it is found in the nucleus and also mitochondria, but mainly in the nucleus. DNA is found in mitochondria as well as the nucleus. Please understand that. Over here, in the case of RNA, it is found in the nucleus and also in the cytoplasm of the cell. Stability, more stable, resists degradation. There it is less stable and degrades quickly. Fine. So we studied this particular thing. Before that we studied what is the genetic material. Okay, what is the genetic material? Then we came around to discuss that what was the first genetic material, and then we can discuss now that what is the better genetic material. So we ultimately got to know it's DNA. Now we are going ahead with DNA replication. So DNA replication, after basically what happens in DNA replication is that there is double stranded DNA. Right. Now in this double stranded DNA, both of the strands are going to separate like this, and a new strand is going to come here. So in this one is the parental strand, this is also the parental strand, and this is a new combination. So if you see, half of it is the parental strand, here also half of it is the parental strand. This method of replication is known as semi-conservative DNA replication method, and this was put down by Watson and Crick, the people who spoke about the double helix structure of the DNA. Will you remember it? Yes. Now ma'am, I cannot believe you. Obviously, don't believe me. Let's have an experiment. So this experiment was again done in the year 1958. So after Watson and Crick spoke about the double helical structure, after that the Michelson and Stahl experiment came. So Hershey and Chase, everything was done before. This is somewhere very, very late. So, okay, so here they explained about the semi-conservative mode of replication. I'll tell you how. So E. coli, my dear children, Escherichia coli, was grown in a medium that was containing N15 isotope. N15 isotope. Now obviously this N15 will be picked up. E. coli has the DNA, and nitrogen is a part of the DNA, nitrogen is base. So the nitrogen which is incorporated will be what? Of 15 type, N15. So the DNA would be having what? N15, 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 right? It will be having N15. Now if you try to centrifuge the whole thing in a cesium chloride medium, it's a density gradient medium, okay, things will arrange itself in the form of density. You will find that this DNA which is there, it will settle somewhere over here. It's heavy. It is what? Heavy. Because both the strands have what? N15 isotope of nitrogen itself in their DNA. Now, I changed the medium. I put N14 isotope. Got N14 isotope in the medium. Now, the E. coli which is there, it will divide itself obviously. Okay, it will do reproduction. And now obviously whenever you're doing that reproduction, you make sure that the DNA that is there, you're replicating and you're giving both the babies the copies of it, right? Both the progenies the copies of it, to be precise. In that case, my dear children, again you would have to make a new strand. We studied the parallel strand is there, that is okay. The new strand will definitely take the nitrogen from the new medium, that is N14. So here the new strand which is formed, my dear children, would be N14, and the older one will be what? N15. Obviously he cannot see it. Again he centrifuged in the cesium chloride medium, and then what he found? This particular thing was somewhere in the middle, in the density gradient medium, somewhere in the middle this particular band was being formed. That means it was hybrid. That means you're getting one copy here. That is why you have 115 and 114. Are you getting it? And now again, always remember E. coli takes 20 minutes time to divide, or 18 minutes to be precise, okay? So again after 20 minutes, that is from the starting point, if you calculate 20 plus 20, it will be 40. Still you are growing it in what? N14 medium itself. Now my dear children, both of them will divide. One new strand will be coming up. So here both the strands will now become N14. Here the red one is still there, the new one will be N14. Here the red one is still there, the new one will be N14. Again here the green one is still there, new one will be N14. In that case, two of them are pure N14, the one that we didn't see until now. So this one is forming another band which is light band, and this one is what? Hybrid. With this, what he conclude? That yes, it is actually replicating in the form of semi-conservative method, right? Which is why you have one parallel strand, which is why you're still seeing the hybrid. Otherwise you would not have seen this. It would be all N14. Now whatever generation you do, my dear children, anyway you will find the hybrid. Why? Because this is there. Anyhow, whenever it replicates, the one strand will be always there. You get it? So this was Michelson and Stahl experiment in the year 1958. Was this the only experimental proof? No. There is one more experimental proof done by Taylor and the colleagues in the year 1958. Okay, and they used radioactive thymidine. They used what? Radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes. Okay, and this was performed on Vishya Fava. Now this question is very much important, which is why I am discussing. Okay, there can be an MCQ question, or if at all you are someone who is preparing for NEET or KCET, my dear children, this question can come in. Taylor and Swift, they also tried to prove the semi-conservative method of replication with the help of what? Vishya Fava, which is this. Faba beans, you also call this as Faba beans. In the year what? In the year 1958. Done. Cool. Understood? Okay. And what did they use? Radioactive thymidine, nothing else but radioactive thymidine. And thymidine is also a part of DNA. Okay. Now, which enzyme does the DNA replication? So we studied about, okay, replication is happening, semi-conservative replication is happening. But what are the enzymes which are being involved over here, and what is the whole thing? So I'll start off with the second thing first, and then we will go ahead over here. This thing. See over here, my dear children. Or should I explain you first? I'll explain you first. Listen to me carefully, okay? The DNA which is there, suppose it's like this. Agreed? So this is 3 dash and this is 5 dash. This is 5 dash and this is 3 dash. I'm going to open it up. Now it looks like this. 3 dash, 5 dash, 3 dash, 5 dash. So first of all, the bonds over here have to be broken. Who does that? It is the work of helicase. Helicase does that, and let me tell you, since hydrogen bonds are very tightly packed over here, it needs huge lot of energy for this enzyme to break the bonds, which is why this process is energy using. Please remember it. Okay, now that is done. Helicase unwinds it. Now obviously these are single strand, they will again come and bind. To stop them from binding, you need some proteins which comes and attach over here, which are known as single stranded binding proteins. Easy to remember, single stranded binding proteins. So they do not allow them to come together back again. Now if you want to make a copy of the DNA, always remember the enzyme which is there, it is known as DNA polymerase. What is it? DNA polymerase. And it says that I will work only in 5 dash to 3 dash direction. What direction? 5 dash to 3 dash direction. So if at all the DNA is in this direction, 3' to 5', then it's totally fine, because why? The DNA polymerase, obviously here antiparallel strand should be formed. So the antiparallel one will be 5' here and 3' here. So it's all cool. For this particular strand, the process is really cool. Okay. Now I call this strand as leading strand, since everything is very cool over here. And one very important thing before I go ahead with DNA polymerase is that this guy cannot start ahead very easily. It needs the help of a primer. Okay, it needs the help of a primer first. So first a small segment has to come and attach, and one enzyme is responsible for that. It makes the primer up over here, and now DNA polymerase will extend this from what? From 5' to 3' direction. That is done by DNA polymerase. So, but first the primers are definitely needed out here. Okay, now what about this strand? This strand is in 3' to 5' direction. So 5 to 3 would be what? From here to here. From here to here. For that you need a primer. Okay, I'll attach a primer over here, ma'am. But now what happens? When the DNA will open more, what will happen? Again you will need a primer, because there should be someone. Again this strand should be elongated from DNA polymerase, my dear children. So in between you have the primers. So do you think the process of replication is smooth over here? No. Which is why you call this strand as lagging strand, because it lags. And the fragments of DNA which is being formed over here are known as Okazaki fragments. What do you call them? Okazaki fragments. Ma'am, then what happens later on, ma'am? Basically, after the replication is being done, this slowly the primers are being removed. This particular parts which are there, they are being removed, and then, my dear children, DNA polymerase would extend, because anyway it has got a tail now from 5' to 3'. Now DNA polymerase will extend this. First of all, an enzyme comes and remove this. Second, DNA polymerase will extend this, and then there will be small gaps. Those gaps are filled by the other enzyme which is known as DNA ligase. Ligase means they ligate things. Okay. And this place where all the replication is happening, it is known as replication fork. Fork, as in it looks like a fork, no? Fork, it looks like this. Similarly over here also it looks like a fork. Suppose replication is happening from that side, replication is happening from this side also. When both the replication forks comes together, it stops there. Replication would be stopping there. You need to know that. That is how it can terminate. Or it can directly go up to some sequences which are known as terminator sequences. So first helicase came, cut it open with the help of ATP, and then single stranded binding protein came, made it stable, and then primer came, my dear children. Primase enzyme came and put primers over here. DNA polymerase came, extended this. Okay, here it is all fine. On the other side, again primase came, put the primer over here. Again primase came, put the primer over here. And then later on, once it was formed, the primers were removed with the help of an enzyme. DNA polymerase extended this, and then DNA ligase joined it together. This is the process of DNA replication. And always remember, replication does not start at any site. It is very specific. DNA polymerase will start the replication from a site which is known as origin of replication. What? It is known as origin of replication. Okay. And where do they get the energy, as I told you? So there are deoxyribonucleoside triphosphate. So deoxyribonucleotide triphosphates, as in this is what you call ATP and things like that, they are acting like that. Triphosphate, as in 3 phosphate groups are there. Okay, 3 phosphate groups are there. So in this pentose sugar. So what happens is that when the phosphate bond over here is being broken, there are so much, 3 phosphates are together. If the bond is broken between them, there is so much amount of energy being released, and that is something that drives the whole process. So energy sources, what? This thing, and they are very accurate. Now we will see this particular video. See, helicase would come in, it will open up. Okay, single stranded proteins are not there. Primase would be coming in. DNA polymerase would be extending it. Over here, again you need a primer. Whenever you open this, again you need a primer, but here everything is going on smooth. Okay? Got it? Same thing happens. So you see Okazaki fragments. Okazaki fragments are over here. And it goes on. Okazaki fragments, my dear children, and later these primers are also being removed. That is not being shown over here, but anyways, Okay, so this is the step. Now we will read about the whole thing. What is happening over here? So the enzymes involved is DNA dependent DNA polymerase. That means you need a DNA to make another DNA. That is DNA dependent DNA polymerase. This enzyme works only on the DNA and it needs a DNA to make another DNA. Catalyzes the polymerization of deoxynucleotides using a DNA template. Efficiency, if we talk about that, in E. coli, where the number of base pair is 4.6 into 10 to the power 6, we have 6.6 into 10 to the power 9. This is 4.6 into 10 to the power 6, my dear children. Completes replication in 18 minutes. That means the average rate of polymerization for making a polymer is what? 2000 base pairs per second. Again an MCQ question. 2000 base pairs per second it is being joined. You imagine how fast it is. E. coli finishes replication within 18 minutes. The average rate of polymerization, if at all you are asked, it is about this only, because in NCERT nothing else is given about any other eukaryotes. So you have to consider 2000 base pair per seconds. For eukaryotes it's obviously different. Now DNA polymerization occurs from 5' to 3' direction. DNA polymerization requires the origin of replication. Accuracy, it is highly accurate. High precision is required to prevent what? Mutation. Energy source, deoxyribonucleoside triphosphates acts as both substrate and energy providers. Why substrates? Because, my dear children, the same as I told you, they are energy providers and substrates also, because over here triphosphate, 3-phosphate is there. When I break this bond, I get energy, and the same thing if I remove the 3-phosphate and I have nitrogen base over here, this is just like that. I am just removing the phosphate group, this gives me energy. Rest of the thing I can use it for forming what? For forming the nitrogenous base out there. You get it? Which is why they are acting as a substrate and also the energy providers. Now going ahead, what other components do we need? Origin of replication, I told you, DNA helicase unwinds the DNA, DNA dependent DNA polymerases, replication fork, DNA ligase that joins the things, and DNA replication occurs in the S phase of cell division. Remember, in the 11th grade you have studied, there is M phase where the mitosis, meiosis, everything happens. In S phase the DNA gets duplicated or replicated. Okay, S phase, synthesis phase. Now, DNA replication, I already told you there is initiation, elongation and termination. Initiation is when DNA helicase unwinds the double helix at the origin of replication. Then single stranded binding proteins keeps the strands apart. Primase adds the RNA primers to start replication. You need RNA primers itself. Later it is being removed, don't worry. Elongation. DNA polymerase adds new nucleotides in 5' to 3' direction. Leading strand is made continuously. Lagging strand is made in Okazaki fragments. DNA ligase joins the fragments together later on after removing the primers. DNA ligase do not remove the primers, there is another enzyme to do that. Now termination. Replication stops when two forks meet or when they reach the specific termination sites. This is about DNA replication. Done? Dusted? Clear? Children, are you understanding this thing? So from 1 hour I am speaking continuously to make you understand certain things, and if at all you are understanding, you have to let me know that. Okay, I'll be so happy to read down your comments. I'll be so happy to solve your doubts as well. So please do that. Now, we studied DNA replication, ma'am. DNA has been duplicated. But how does this DNA form the protein? You told that is some RNA being formed, then RNA is transformed into some proteins or something like that is happening. Yes. So how does DNA help in the formation of proteins? For that, you should remember the central dogma. According to the central dogma, the one that we didn't study, okay, the one that was before, before the histone proteins and everything, the nucleosome structure, that is this. According to central dogma, it says that the DNA which is there, you need to make a copy of it. This is known as replication. Agreed? The DNA to form proteins, in between you need to form RNA, and this process is known as transcription. And then the RNA transfers this information to form the protein, and this is known as translation. This was given by Francis Crick. Central dogma was proposed by Francis Crick. Double helix structure was given by both Watson and Crick. So over here, the gist of the story is that this is the DNA. You form the RNA out of it. The DNA stays intact only. You just make an RNA by the process of transcription. This RNA will move out of the nucleus. Everything happens in the nucleus, right? This RNA will move out of the nucleus. Out of the nucleus, in the cytoplasm, they will meet ribosome and also tRNA. This is another type of RNA, tRNA. both of them will help in making proteins out of what? The mRNA's information. So mRNA is not transforming into proteins, mRNA is helping in the formation of proteins, and that process is known as translation. And we are going to see each of them one by one. First is transcription. What does the transcription unit contain? So this is a transcription unit. So as you can see over here, transcription unit contains a promoter region, the structural gene, and a terminator region. The upper strand is known as template strand. Here the lower one is known as coding strand. As in, it's not upper and lower, you have to consider the polarity. The one that is in this direction, okay, 5' to 3' over here, this would be a template. The one that is going to undergo transcription is known as template strand, and the one that does not is known as coding strand. So if at all you are asked, ma'am, where is the promoter region, you say towards the 5' end of the coding strand. So this guy is not involved in transcription, you need to give it something, right? Otherwise it will be sad. So 5' end of coding strand, you have the promoter region, and 3' end of the coding strand, you have the terminator region. Now why do we have promoter, terminator, everything? Because here also there is an enzyme that helps in the formation of RNA, and that enzyme's name is what? RNA polymerase. Okay, its name is RNA polymerase. The RNA polymerase specifically comes and binds only on the promoter region, and it does that when it combines with its friend that is known as sigma factor. So the RNA polymerase gets combined with sigma factor, and now these guys will come and attach on the promoter region. Now you can guess in which direction to which direction things are forming. This is 3' to 5', so it will be forming in 5' to 3' direction itself, just how DNA is being formed. Only difference is that in the case of DNA, if at all you have thymine, in the opposite complementary strand you will have adenine, right? Agreed. Or in the DNA strand, if at all it is adenine, in the opposite strand it will be thymine in the DNA. But over here, if it is adenine, then here the RNA will have uracil instead of thymine. Okay? That's the difference that you will be finding over here. So polymerase plus sigma factor comes and attaches to this promoter. Without sigma factor, can it come and attach? No, it feels so shy. Sigma is for making it not too shy. You remember like that. And then my dear children, later transcription will happen in 5' to 3' direction. It wants to terminate. Now it feels like, okay, I have formed an RNA, I want to move, I don't like it. It's RNA, I am an RNA, I don't want to be within DNAs. So it is crying. And what do you call crying in Hindi? Rona, Rho. So Rho factor will be coming in and saying that I can save you. So the Rho factor comes in and helps in the termination process of transcription. So sigma factor over here to avoid the shyness, to make friends, and Rho factor to save it later on. There might be some regulatory sequences over here and here. That means regulatory sequence, on the regulatory sequences, certain things can come and bind, and when they come and bind, the RNA polymerase won't be able to move ahead. A lot of things can happen like that in general. Okay. So if at all you are asked to make a transcription unit, my dear children, you need to understand you have to mention promoter, terminator. If at all you are mentioning this as the template strand, then your polarity should be 5' to 3' direction, and the other one would be 5' to 3' in the opposite way. Okay, RNA polymerase always works in 5' to 3' direction, as in it makes the new one in 5' to 3'. And as I was saying, initiation, elongation, termination here also. RNA polymerase binds to the promoter with the help of sigma factor. Okay, the DNA helix opens and the transcription begins. Elongation, RNA polymerase add nucleoside triphosphate. I told you they are acting as a substrate and also as energy givers. Triphosphates, in that 2 of the phosphate goes out, only 1 phosphate remains. That forms a nucleotide, forming the complementary base pairing, only the short stretch. Okay, anyways. Termination, RNA polymerase reaches the terminator region. Rho factor helps in termination, causing the RNA to fall off. RNA polymerase detaches, completing the process of transcription. Again, initiation. You have to remember the sigma factor and Rho factor. That is very, very important. Now, there is a difference between prokaryotic transcription and eukaryotic transcription, my dear children. Prokaryotes means the ones that do not have a proper membrane-bound nucleus, that do not have membrane-bound organelles. Okay, so that are prokaryotes. They are primitive in nature. For example, bacteria. Eukaryote as in us, as in the plants, everything. So if at all you are asked, pea plants, what type of thing do they have? And you have to tell that they have monocistronic things. Okay, they are monocistronic in nature. What do you mean by that? I'll tell you. Polycistronic means They have multiple structural genes that codes for multiple protein. That means if at all I think about the sequence, over here I have promoter, over here I have the terminator. In between that there will be multiple structural genes that can code for A, B, C proteins out there. But when you talk about monocistronic, over here this is the promoter, this is the terminator. In this region there will be only one gene coding for one protein. One gene will be coding for one protein, but over here it's not the case. That is what is known as polycistronic. Transcription is happening over here in the case of prokaryotes, as in bacteria and things like that. Eukaryotes, as in us, as in plants, they have monocistronic. They're very specific. Next, prokaryotes do not have introns. What do you mean by that? So from the DNA, you are going to make the RNA. Now the DNA has certain regions which do not code for anything, such as this region. They are not coding for anything. There are regions that codes for proteins. There are regions that does not code for anything. The region that codes for proteins are known as what? The region that codes for protein are excellent. That is why they are known as exons. Okay, and there are regions that does not code for anything. They are in between the exons. They are known as introns. Okay, in the case of prokaryotes, in their RNA, they do not have introns, my dear children. Okay, they do not have introns at all. But in our case, we definitely have introns, and it has to be removed later on with the help of processing. So after RNA is being formed, certain processing is also being done. So that has to take place in them. So eukaryote genes are split containing exons coding sequences that appear in the mature RNA, introns non-coding sequences. I'll talk about that, don't worry. Transcription and translation occurs in the same area, that is cytosol. That means here, since there is no since there is no nucleus and everything, my dear children, so transcription, translation, everything happens in the cytosol itself. Whereas when you talk about eukaryotes, we have a separate nucleus and then we have what? Cytoplasm. So I told you transcription happens in the nucleus, but translation happens outside because ribosomes and tRNA are outside. So it happens in the nucleus and half of it happens in the cytosol. Please remember it. Okay, so these are the three things that you need to know: polycistronic, monocistronic, but they do not have introns. We have introns, and those has to be removed, and we are going to talk about that over here. So I'll talk about this first, then we'll talk about that. Okay, RNA processing. So once the RNA is being formed, always understand, when I say that DNA helps in the formation of RNA, the first RNA which is being formed is known as heterogeneous nuclear RNA, hnRNA. Very important question. And as I said, it has exons, it has introns, exons and then introns. And then it undergoes processing, it undergoes some changes such that it finally forms the mature RNA, which you call as messenger RNA. This is not for mature, this is for messenger RNA, the one that carries message outside the nucleus. This messenger RNA will not have intron regions because that has been cut out, it just has the exons out there. This whole process where certain things are being changed out here, it is known as RNA processing, and is very much important for making the the process very, very smooth. So first such process is splicing, where it removes the introns. Splicing is the process by which we are removing the introns. Next there is a process that is known as capping. In capping, what we are doing is that we are going to add methyl guanosine triphosphate at the 5' end. So this is the RNA. And the 5' end, we are going to add methyl guanosine triphosphate. There will be 3 phosphate group over here, my dear children, and this is methyl guanine triphosphate. Why do we add it? Because, my dear children, there are certain enzymes that can come and cleave this particular part. And we do not want that. We want to keep it safe because this is the part that will be giving rise to what? Proteins later on. So we want to keep it safe. Similarly, on the other side also we have to keep it safe, my dear children, which is why we do the other process that is known as tailing. So here it is capping and there it is tailing. What do we do over here? We almost give 200 to 300 adenylate residues at the 3' end. Adenine group are put up, my dear children, so many, 200 to 300. So even though cleaving happens, it will happen slowly and this part will be again safe. So splicing first, where you are removing the introns. Capping, where methyl guanosine triphosphate, GPP. So it is written like this. Methyl guanosine triphosphate, 3 phosphate group. okay, is being added to the 5' end, entailing 200 to 300 adenylate residues is being put at the 3' end. And the fully processed mRNA is then later transported out toward the cytoplasm for the process of translation to happen. And then tada, the story is over. Cool. So here the same thing is being mentioned. Ayyo. Okay, so over here you get the RNA, you are doing the capping so that it's safe, and now my dear children, you have the exons, introns and everything, you do RNA splicing, and then you are removing the introns are not there. With the help of splicing, you see, introns are being removed, and then you just have the exons, and at the end you will do polyadenylation also. Here you can see polyadenylation, just once it is being removed like this, polyadenylation and splicing is also happening. Initially capping is already being done because there's still one end is still free. That is why capping is being done. Once it is removed, release my dear children, splicing and adenylation is done, polyadenylation is done, and then you tada, you get what? mRNA finally. Now the thing that you need to understand is that in our case, since everything is very much complicated, we need different types of RNA polymerase, which is not actually found in the case of prokaryotes. So we have 3 RNA polymerase. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and you can remember it like this. Robo 1 reads, that means RNA polymerase 1 is responsible for rRNA formation. rRNA is what? Ribosomal RNA. Ribosomal RNA. This will be helping in the formation of ribosome, which is an organelle. Okay, so these rRNA together helps in the formation of ribosome later on. So RNA polymerase 1 transcribes what? rRNA. RNA polymerase 2, which is there, it transcribes hnRNA. The formation of hnRNA is being done by that. Okay. RNA polymerase 3 transcribes tRNA, that is transfer RNA. Transfer RNA is going to help in the process of translation. You are going to study about that. And also 5S rRNA. So one rRNA is different, that is 5S rRNA, it will be over here. And also small nuclear RNAs, that is also transcribed with the help of RNA polymerase 3. That means they are all formed with the help of RNA polymerase 3. So Robo 1 reads, read stands for rRNA. Robo 2 messages, okay, mRNA. Robo 3 transfers, that means tRNA as well as small nuclear RNA. Small nuclear RNA, and one exception is there, that is 5S rRNA. You can remember like this is very much important, and the subunits, my dear children, are different: 28S, 18S, 5.8S, all of them has 8. 28S, 18S and 5.8S. So these RNA polymerases are different in the case of eukaryotes, since we are what? More complex in nature. Now going ahead with the next thing, that is the genetic code. We studied about the transcription. Now for translation, we should know what are certain codes in the RNA, in the mRNA that we formed. There are certain codes, and we should know what is the meaning of those codes. So over here, when I say that this is the RNA, and I am saying that AUG is there, UUU is there, UCU is there, or UAA is there. Now I am pretty sure this particular codon which is there, they are codons, okay, the 1, 2, 3 of them come together, they form triplet codon. This codon is for an amino acid that is known as methionine. UUU is for another amino acid that is known as phenylalanine. UCE is for another amino acid that is serine. UAA is the one that stops everything. That means what, my dear children? Each codon which is there, it is specific for one amino acid. And if at all you know, proteins are made up of amino acids, and I told you mRNA is going to help in the formation of proteins. So mRNA has that information as in what amino acids have to be made, and these amino acids comes together to form what? Proteins. And there are certain unique features of genetic code which you should know. These are known as genetic code, and you should know the features. First one, the codon is triplet in nature, that means 3 of them come together. 61 codons code for amino acid, 3 codons do not code for any amino acid. Okay, so total 64 are there. Total 64 are there. 61 of them are coding for amino acid, and 3 of them are not coding for anything. They are known as stop codons, the one that will stop the process of translation. So I'll come directly to the 3 of them which are stop codons, because in that way you will be able to study it easily. It is UAA, UGA and UAG. UA, UAG, UGA. Okay, UA, Uga, uag. Either way you remember it. So you say whenever something burns, you say uah, okay, and then wait. Okay. Uga is there, you have to remember uga. I hope you can. And then you saw fire, so you say uag. Okay, ag means fire, right? And then ah means you went to touch it and then you got hurt. Either way you remember it, but you please remember it and do not tell this to anyone else because they will beat you. Okay, so these are the 3 stop codons, but you definitely need to know about them. And my dear children, so yes, 63 of them code, 3 of them do not code, and those are this. Now some amino acids are coded by more than 1 codon, and hence the codes are degenerate in nature. What do you mean by that? That means over here, this amino acid, serine, they are coded by UCU, UCC, UCA, UCG. Each codon is coding for a amino acid, but all of them coding for the same amino acid, my dear. So this amino acid have multiple codons. So this feature is known as degenerate. Some amino acids are coded by more than 1 codon. This is known as degenerate feature of the codon. The codon is read in mRNA in a contiguous fashion. This is another feature. So over here, do you see any punctuations, full stop, comma, anything? No, it is read completely like that. That is contiguous fashion. Now the code is nearly universal. For example, if you talk about, if you put the same mRNA in the case of bacteria also, they are going to produce the same protein. That means what? The codon is read in the same way there also. So the code is nearly universal. For example, from bacteria to human, U would definitely code for what? Phenylalanine. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. So mitochondria, the whole different world is happening. There you can see some exceptions, and also in the case of protozoans, the primitive ones. Protozoans are eukaryotes, but the primitive eukaryotes. Okay. Now one more thing is there. AUG, which is there, it is coding for methionine, and it is the one that is start codon. It has dual function. It codes for methionine and it is also a start codon. If it is there, then only the process of translation will happen. UAA on the other hand, they stop codons, they do not code for anything. Translation will stop here, but it has dual function. So these are the features of genetic code, and the genetic code was given down by George Gamow. His name was what? George Gamow, my dear children. Now going ahead, there are several scientists who came into the picture. Severo Ochoa, who said discovered an enzyme polynucleate phosphorylase that synthesized RNA without needing DNA. So he made an artificial RNA without the DNA. Usually we have a DNA, then it gets transcribed to form an RNA, right? But he artificially made that. Okay, he artificially researched about it. Hargobind Khurana, he chemically synthesized the RNA sequences. What did he do, my dear children? Okay, wait. Sorry. Severo Ochoa, he was the person who helped in the formation of the enzyme that would help in the formation of an artificial RNA. Now, who made the RNA? It was Hargobind Khurana. He picked up the bases and he made the RNA using the enzyme that the other guy created. But I know that I have put this much bases, A, U, G, everything. Now, Marshall Nirenberg came into the picture. He developed a cell-free system to experimentally confirm which codons code for which amino acid. So inside, it's not happening inside the cell, but outside the cell, my dear children, outside the living cells, he was able to make this codons which were there code amino acids. And with that, if at all, my dear children, if at all, I already know that this is the codon that I have put. And now I get to understand that this codon is coding for this particular thing, that is methionine. So I would assign AUG for methionine, right? So the whole thing was done by these 3 guys out there, and you need to know about them. Severo Ochoa, Hargobind Khurana and Marshall Nirenberg. These 3 teachers, or what do you call, scientists, are really, really important for you guys. As in, just for understanding, it's not for remembering. You don't need to remember much. Hargobind Khurana and Marshall Nirenberg is something that you may remember. It will be good. This is an extra thing. Okay, now we studied about transcription also. Before we go ahead with translation, there is a thing that I need to discuss, and that is mutation, my dear children. So insertion or deletion of 1 or 2 bases changes the reading frame from the point of insertion and deletion. This is known as frame shift mutation or deletion mutation. So I want to give an example because on this numerical question comes in. So Ram has red cap. That is the statement. If I put 1 nucleotide extra, the whole reading frame changed. Ram has decap. it becomes 333 if it is read different now. So this type of mutation which is there when I am removing one or I am adding one, this is known as frame shift mutation. What mutation? Frame shift mutation. On the other hand, if I included 3 of the other things, for example, Ram had red cap is the initial statement. Now instead of putting 1 or 2, I put 3 of them together. For example, SAD, S-A-D. In that case, is the reading frame changed? No. Just that one more amino acid will be formed. That's all. Now, suppose my dear children, this nucleotide is UAA. So then, transcription will happen from here to here and it will stop. So it can be depending on that. Whatever nucleotides are being added up, if at all it's 3, anyway frame shift mutation is not going to happen. But the next thing that can happen, if it is all a stop codon, my dear children, if at all it is a stop codon, the whole process of translation, not transcription, translation would be stopping over here. If it is not a stop codon, translation would be happening with an additional amino acids over here. Okay, please remember it. Now, coming to the next thing, that is what? Translation, the next process. What exactly happens over here and what do we need over here? So, we need tRNA, that is soluble RNA, or you call this as sRNA. Initially, it was known as soluble RNA. I told you tRNA is found outside. tRNA is made with the help of RNA polymerase 3. In tRNA, again it's an RNA, so it will have a 5' end and a 3' end. On a 3' end, my dear children, amino acid comes and attaches. And this process is known as charging of tRNA. What? It is known as charging of tRNA. Okay, or you call this process as amino acylation. This happens in the first phase of translation. Remember, this is not the first step. This is happening in the first phase of translation. Translation has officially not begun. Okay, and always remember, tRNA will have this loop which is known as anticodon loop. What happens over here, ma'am? So, anticodon loop will have a set of nucleotide bases. And as I told you, this tRNA is attached to an amino acid. Now, suppose over here in the mRNA it is AGU. The tRNA which has the complementary base pair to it will be coming and attaching over here, right? And that will be coding for one amino acid. Similarly, all of them are red. So, always remember the 3' end of the tRNA is the amino acid acceptor end, and amino acid comes and joins with the help of charging of tRNA or amino acylation, and this is happening in the first phase of translation, and this is the anticodon loop. Now just give me a small second. Okay, now one more very important thing that you need to remember is secondary structure of tRNA has been depicted that looks like the clover leaf. Remember, tRNA has a secondary structure as well that was said to be looking like a clover leaf, but now it has been said that the actual structure tRNA is compact molecule which looks like inverted L. So previously clover leaf, but now it is known as inverted L model. Please remember it. Okay. Now it is also known as soluble RNA, sRNA. Don't get confused. It's not small RNA, it is soluble RNA. Now, my dear children, tRNA charging happens in the first phase. Amino acids are activated using the ATP and So, whenever charging of tRNA happens, whenever amino acids are being joined, please understand it needs a lot of ATP. It's a highly energetic process. Okay, charged tRNA facilitates peptide bond formation. Okay, we'll come to that. Now, ribosome structure and function. Ribosome which is there, it is known as the protein synthesizing factory. Why? Because over here ribosomes has a larger subunit and a smaller subunit, and in between that sandwich is what? Your mRNA, and this mRNA is now going to be read by the tRNA out there. So, since they are helping in the formation of what proteins, they are known as the protein factories. It exists in two subunits, large and small, in an inactive state. The large subunit has two sites for amino acid binding. So, it will be having two sites where the tRNA will be coming over here, and the binding will be happening. So, suppose over here amino acid there, here also amino acids there, both of them are going to get bonded with the help of peptide bonds. Amino acids are joined by peptide bonds requiring energy. The 23S rRNA in bacteria acts as ribosome to catalyze the peptide bond formation. I told you RNA can act like a catalyst or an enzyme. So, over here rRNA which is there, which is a material that is helping in the formation of ribosomes, not a material component that helps in the formation of ribosome, it is also helping in the formation of what? Peptide bonds out there. So, in the case of bacteria, you have a name to remember, and that is 23S rRNA. 23S rRNA in bacteria acts as ribozyme, that means an enzyme that catalyzes what? Peptide bond formation. Okay, now one more very important thing, whenever you talk about translation, whenever you talk about mRNA, there are certain regions before the 5' end and after the 3' end that does not get translated, and this is a very important question. Okay, and what do you call these regions as? You call these regions as untranslated regions. They are untranslated regions, UTRs. Okay, also you call it UTRs. Why it is there, my dear children? To protect the mRNA, to make it stable, okay, to help in the process of translation, to protect, to provide stability, and for what? And for helping in the process of initiating the process of translation. Always remember, protection, stability, and helping in initiation of translation process. Will you be able to remember it? And it is before 5' end and it is after 3' end. Okay, done? Cool. Now, translation process, initiation. The ribosome binds to the mRNA at the start codon recognized by the initiator tRNA. Okay, it is recognized by the initiator tRNA. Fine. The ribosome binds to the mRNA at the start codon recognized by the initiator tRNA. Okay, first of all, I'll tell you, rather than following this, you can just listen to me carefully first. Okay? Okay. So first thing that happens is the smaller subunit comes. First point. First phase is done, that is charging of tRNA. Now first point is that the mRNA is going to bind with the smaller subunit. Okay, done. Second point, so this is the first point. Second point is that the tRNA, initiated tRNA will come and bind over here with the amino acid, that is methionine. And over here you have AUG, then there it will be UAC, right? Now my dear children, the larger subunit will be coming in. So this is the second step, and now this is third step. Larger subunit will be coming in. Now again a tRNA would be coming in. Later this amino acid and this amino acid is going to form the peptide bonds, and then when they form the peptide bond, what will happen, my dear children? Okay, I have the picture over here. When they form the peptide bond, here this and this links, the tRNA goes off. So now you have a strand of what? Amino acids up there which are linked through the what do you call this as? Peptide bonds. Okay, and now when it reaches the last stop codon, it will stop the process of translation. No other tRNA will come over here and bind. Okay, instead what will come and bind? A release factor will come and bind such that the whole polypeptide chain goes off free. So termination happens like that over here. Elongation means amino acid containing tRNA will come, and now they will form bonds with the help of the enzymes out there. Okay, and later in the case of termination, a release factor binds to the stop codon, freeing the polypeptide which is being formed, and that is how translation also gets completed. Now, since we have studied about replication, transcription, translation, now you tell me, gene regulation can be controlled at what levels in the case of eukaryote? That means, if there is a DNA and I want to make protein, or in other words, I do not want to make proteins, in what all ways can I stop? In what all ways can I stop the whole process to happen? First thing, transcription level. I would not allow transcription to happen. The formation of primary transcript, that is formation of H and RNA, I can stop that. So that level I can do things. Second thing, suppose H and RNA is being formed, then I'll make sure the processing is not happening to form mRNA. So at processing level I can stop. Third thing, okay, mRNA is also being formed. I can stop mRNA from going outside to the cytoplasm. So transport of mRNA can be stopped. Now fourth thing, translational level. That means this mRNA, if it has moved outside also, I would not allow translation to happen. So in that level, in these 4 levels, my dear, again a very important question, in these 4 levels, I can control the gene regulation in the case of eukaryotes. Now what about prokaryotes? How can I control the gene regulation up there? So in prokaryotes, things are quite different. Okay, in prokaryotes, control of the rate of transcription, since they do not have different regions, nucleus, cytoplasm, this and that, everything happens in the same region, which is why you have different ways to control gene regulation out there. Control of the rate of transcription initiation is the predominant site for gene expression. So only thing that you can do is, when the transcription initiation happens, you can make it stop somewhere. You will not allow the transcription to happen. How, ma'am? I didn't get you. I'll give you an example. This is lac operon. Okay, this is lac operon. And over here, listen, properly. This is the promoter region. That means there will be a structural gene. Yes. So this is a structural gene which is known as regulatory gene. A gene is responsible for making a protein, and thus protein is repressor protein. Okay. DNA, RNA, and then protein. Repressor protein is being made. Similarly, you have promoter region for another structural genes out there. Z, Y, A. These are three structural genes. Z, Y, and A. They have promoter region. Apart from that, there is an operator region, and this is going to help in regulation of what you call the expression of the gene. As I told you, in the case of prokaryotes, things are quite different. So there is operator region also. What happens is that this is of E. coli, and the E. coli is grown in a medium which does not have enough amount of lactose. Okay, lactose is not there. Their energy source is not there. In that condition, this repressor protein is being made, and this repressor goes and binds on the operator region. Okay, now ma'am, what's the problem in going and binding the operator regions? The RNA polymerase, when it comes and binds to the promoter and wants to do the transcription, will it be able to do it? No, it will get stopped. It will get stopped. So if transcription of these three genes are not happening, my dear, that is a problem, right? So here, this condition is known as switch off condition of lac operon. Switch off. Now suppose I am growing the same thing in a medium that contains lactose. What happens then? The lactose comes inside the cell. The lactose sees that, okay, someone is binding on the operator region and my friends are not getting transcribed. So lactose goes and binds with the repressor. Lactose goes and binds with the repressor. Lactose is known as inducer. I'll tell you why. Lactose goes and binds the repressor. Now the operator region is free. RNA polymerase can come attach and do the whole transcription. Transcription will happen. Z will code for beta galactosidase enzyme, Y will code for permease enzyme, A will code for transacetylase enzyme. Y is known as inducer because it is inducing the process of transcription. It is helping in the start of transcription. So lactose can be there or allolactose can be there. Both of them acts as inducer, and this is the switch on condition of lac operon. This is how things are regulated in the case of prokaryotes. Now, what does these enzymes do? So the lactose which is there, I told you, it's an energy source. So beta galactosidase which is being formed, it actually breaks down the lactose molecules. So don't think that only one lactose is there will be much molecules, right? So those lactose molecules are broken down to glucose and galactose, so that it gives the energy. Lactose is broken down to glucose and galactose such that it gives energy. Permease enzyme is there that will increase the permeability of the E. coli membrane such that this lactose can come in. So ma'am, in the initial case, how did the lactose come in? Even though we say that this switch off condition is there, but little amount of proteins are still being made, which is why there is little amount of permeability, which is why lactose was able to come in. Okay, transacetylase is the one that will be managing all the byproducts of all these processes out there, and it has other functions which are not being mentioned in your NCERT and which is not needed much over here to you to know. So do you get it? And this regulation is negative regulation. You are negatively regulating it with the help of what? Repressor protein out there. But it is for good. Why do you need to make the proteins when it is not needed? Whenever lactose is not needed, what will it break down? We get it? So yeah, that is the thing. Okay. So over here the whole thing is written. Lac operon was actually found by whom? Where is the cursor? Lac operon was elucidated by Francois Jacob and Jacques Monod. The lac operon is transcriptionally regulated system in the case of bacteria. Regulatory gene is there, that is I gene, which produces a repressor protein. Structural genes are there, that is Z and Y gene, my dear children, and also A gene. Z gene codes for beta galactosidase, breaks lactose into glucose and galactose. Y gene codes for permease, helps lactose enter the cell, and codes for transacetylase. Okay, see A1. Zia, you can remember Ziada, Zia. Now, function and regulation. Lactose as an inducer. In the absence of glucose, lactose enters the cell via permease. The repressor produced by the I gene normally binds to the operator, blocking the transcription. Lactose or allolactose inactivates the repressor, allowing RNA polymerase to transcribe the operon. Regulation type, negative regulation via repressor. Positive is also there, but that is beyond discussion for your level. That is 12th grade level. It is not by me, it is by the NCERT written over there. Now, we studied about everything. Now we'll come to Human Genome Project. What do you mean by that? So Human Genome Project, their aim, what was the aim? To actually find all the genes out there in the human DNA, all the genes. Almost 30,000 were there, but over here 20 to 25,000 is being written. Now after that, they just didn't limit to that. They wanted to see all the base pairs of the whole genome, my dear children. Determine the sequence of the 3 billion chemical base pairs out there. Now once you did that, what do you need to do? You need to store the information in the database. And then if you are storing the information, you need to have tools to find them easily. So improve tools for data analysis. Then you do not keep it with yourself. You are not selfish. You want to transfer this data to some other places as well. These were the aim. And also the last one is to address the ethical, legal and social issues arising from all this thing. You cannot randomly go and kill some organism and find their things. So you have to address all these things. So these were the certain aim of Human Genome Project which you need to remember. Now have any other organisms been sequenced? So yes, non-human model organisms such as bacteria, yeast, Caenorhabditis elegans, that is a free living pathogenic nematode, that was also being sequenced. Drosophila, the fruit fly, plant, rice and Arabidopsis was also being sequenced. Just remember the names. Questions have not come yet, but you never know. Human Genome Project was a 13-year-old project by the US Department of Energy and the National Institute of Health. In MCQs of KCET and things like that, you may find it. Okay. Now, going ahead, how did they actually sequence the human genome? What was the method for finding the human genome, my dear children? So, two main approaches. Express sequence tags identifies only the genes expressed as RNA. So, whatever genes were being expressed, those were being found. That was one way. That is expressed sequence tags. Now, the whole genome sequencing, the other one, sequences the entire genome, both coding and non-coding region, and assigns functions. Okay, sequencing process. DNA is extracted from the cell. It is broken into smaller fragments, and then, my dear children, these fragments are cloned into some bacteria or yeast. Why? Because you want to amplify it, because with one DNA you cannot find the whole thing, so you might need more things, right? So you do amplification, and after that, my dear children, your sequencing was done by automated DNA sequencers based on Frederick Sanger's method. So what was it? What was his method? So over here, the sequences were found out. For DNA sequence, I suppose A, G, C, T, U, oh sorry, not U, C, G and things like that. Again, I got a strand where T, C, G, everything was there. Starting point were T, C, G. So I will basically put this as first strand and I will put this as second strand, because over here I am saving overlapping strands. And then in that way, I am going to see the whole DNA out there. Okay, so this was Frederick Sanger's method, DNA sequencing method. Please remember, DNA sequencing was given by Frederick Sanger. This is a question, match the following question. Was also developed the method for determining amino acid sequences in proteins. Sequence assembly. DNA fragments were sequenced and arranged using overlapping regions. Specialized computer programs were developed for alignment, as I told you, and then the chromosome sequencing was done. Chromosome 1 was the last to be sequenced. Please remember, chromosome 1, which has the highest number of genes, was the last to be sequenced. In May 2006, it was sequenced. Okay, so here the only important thing that you need to know is Frederick Sanger was the one who came ahead with sequencing. Now, what were the findings of Human Genome Project? Again, an important question. So, all these findings, and over here I don't think there is much explanation needed, but still I will tell. The human genome contains 316.64.7 million base pairs. The average gene usually contains 3000 base pairs, but my dear children, there is a dystrophin protein which has around 2.4 million bases. Okay, so everything is in millions. 2.4 million bases. Usually the gene size is 3000, but here it is dystrophin has 2.4. So please remember this unique names, dystrophin. Now the number of genes which were estimated to be too much, it is just 30,000. Only 30,000 genes are there. Now the function of 50% of them are not known. Now, out of that 50% of them also discovered genes only, okay, so 50% of the genes, the discovered genes are the only ones known, others are not known. Now, out of the whole genome, only 2% of the genome actually codes for protein. Okay, it actually codes for protein. Rest are repeated sequences that makes up the large proportion. These repeated sequences are found in human beings in multiple numbers. Okay, this repeated sequences are found in human beings in multiple numbers. In my body, it might be 6 times. In your body, it might be 10,000 times. Okay, but you have it. Now, chromosome number 1 has the most genes. I told you it is 2968. And chromosome number Y has the fewest. Chromosome of Y is found in the case of males, and they have 231, my dear children. 231 genes out there. 2968 and 231. So 231, you can remember like that. So one came late. Okay. Now, scientists have identified about 1.4 million locations where single base DNA differences are there. That means you and me, for example, AGCT. I have this, but you have ACCT. Single base difference is there. This is known as single nucleotide polymorphism. So those were the findings that they did in the Human Genome Project. The goal is different, the findings are different. This is something very important. Please write it somewhere in key points, small points. It might come. Now, the next thing is DNA fingerprinting. Okay, the last topic. So, the total human genome is made up of 3 billion base pairs. How many base pairs? 3 billion base pairs. We have 30,000 genes. The total, 3 billion base pairs, I told you already, right? Now 99.9% of this is identical in all humans, that is you and me. Meaning that is only 0.1 difference. Okay, 0.1 of 3 billion is 3 million base pair difference. This is there. This means that 3 million DNA bases vary between individuals, that is you and me. And that was being used by DNA, that was being used in the DNA fingerprinting method by Alex Jeffrey. So over here, instead of sequencing all 3 billion base pairs to compare two people, scientists focused on specific DNA regions that are different. These variable regions, the repetitive DNA, contain small DNA sequences that repeat multiple times and vary between individuals. So basically, I also have it, you also have it, but the number of times it gets repeated is different. By analyzing these differences, scientists can identify individuals quickly. That means I have 6. Okay, for example, I did a crime. In that crime, the DNA which was there, it also has 6 repeats. I also have 6. I did the crime. So, by analyzing these differences, scientists can identify the individuals quickly. Satellite DNA and polymorphism. This is also given. So, our DNA has bulk DNA, major part, and satellite DNA, small but unique, repetitive sequences. The satellite DNA varies from person to person and helps in the DNA fingerprint. So, these satellite DNA are the one which are the repeats or which is very unique in me and you. Okay, the number of it. Since these sequences do not code for proteins, mutation in them do not affect your health and they can accumulate over generation, making them useful for what? Identification. Now, mostly this part of the story is not being asked. This part of the story is definitely being asked. Okay, so listen to me carefully. Suppose there is a crime that have taken place. Okay, and from the crime spot, blood was there and hair follicle was also there. With this, I got the DNA. I am the police. Okay, I am the police. There is person A suspect and person B suspect, that is you. B is you, A is my friend, okay, and I am the police. Over here, my dear children, I was able to find, when I found the DNA, I was able to find the sequence of it. There were certain sequences which was there. It was AGCT, okay. These were repeated. I got to know that. These sequences were there. Now, what did I do is that first of all, I made a complementary thing for that. So, complementary thing of this is what? TCGA. This is the complementary thing. And I will tag it with a radioactive thing to make it what? A probe. A probe is something which has some radioactive label into it and that is going to help me somewhere. I'll tell you how. So, basically anyway, the DNA that I got from here, I cleaved it with the help of restriction enzymes. Restriction endonucleases are the ones that cleaves within the DNA. Endonucleases. They cut within the nucleic acid, that is the DNA, RNA, whatever is there, DNA over here. Okay. So, I use the same enzyme to cut the DNA that I got from suspect A and the DNA that I got from suspect B. Both of your blood I took and the friend's blood I took. Okay, and I use the same enzyme to cut the whole DNA. And then, my dear children, in the gel electrophoresis chamber, the strands that I cut over here, that was separated. The strands I got from you guys, that was separated. Okay, so from B I got these many, from A this, and this is the criminal. I cannot see it in the electrophoresis chamber. I cannot see the DNA. So, I take a nitrocellular sheet and transfer it over here. This is known as southern blotting. Okay, still I cannot see it. So, I am going to put the probe, radioactive probe over here. Obviously, since it has the complementary stand to something that the criminal obviously will have, so it will only go and bind if at all it's there. So, it goes and binds over here. It goes and binds over here. No, it goes over here, 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 here. Okay, this is criminals, this is mine. Oh, sorry, not yours, the friends. And here it is not mine, suppose. Okay, or, okay, anyways, let's make it bind here also. Here also it is binding. Cool, got it? Now my dear students, still I am not able to say it. I have to use an autoradiography machine to view it. And when I see it, I find that the banding pattern over here and the banding pattern over here is the same. What do I conclude from this? That my friend A is the criminal. Ma'am, why is it going and binding everywhere? I told you. We have certain sequences which are same to same, but the repetition of it differs. In me, it might be 6, but in you, it might be 10. So when the restriction enzyme will come and cut, my dear children, your strand will have 6, 1. Mine would have 10. You get it? Which is why, suppose yours is 10, which is why it is not moving. In a electrophoresis chamber, things move according to the what you call size. So yours is 10, which is why it is over here, but mine is 6, which is why it is over here. Get it? So my strand and your strand would be different, because here I have 6 time repetition, over here you have 10 time repetition. You get it? And then criminals also matches with that. That's when I get to know that he, my friend, is what? The criminal. And this is the process of DNA fingerprinting. First of all, DNA isolation is done, restoration digestion has been done, electrophoresis has been done, okay, separating of DNA fragments depending on the size, and then blotting is being done where transferring of things are being taking place, hybridization is done when radio labeled VNTRs are being used, okay, and then autoradiography is done for seeing the unique pattern. Now in NCERT, one big thing is given over here. So my dear children, see, A and B has this, B has this banding pattern, A has this banding pattern, and C is this. So if you are asked, C is the child, who is the mother, this or this? It will be B. They have almost the same thing. Obviously there will be certain changes. I am not saying there won't be changes, but mostly it would be matching up there. So number of short tandem repeats, this is, so VNTR which is given over here, what does that mean? Variable number of tandem repeats. You and me will have variable number, but the repeats would be the same. Okay, so this is variable number of tandem repeats, and with the help of that we can actually find out things. If at all you need me to explain this to you once more in a separate video, in a small 5 to 10 video, let me know in the comment section below. I'll make a separate video and explain you properly. As of now, there are time constraints because it's almost 2 hours, and according to the timeline, it should not go that far. So my dear children, this was molecular basis of inheritance. I hope you understood it. If at all you think that I have explained to it properly, definitely let me know. If at all you have any doubt, I'm telling you again, you have to let me know about that as well. And I just wish you all the best. Make sure you watch the 1 marker video as well. And the 2 and 5 marker you can download, you can watch it and also download the PDF for getting the answer list. Okay, wishing you all the best. Please take care of yourself. Bye-bye.